This is FX Radio and I'm Andrew Whitfield-Cook, and with me in the studio today is none other than Dr. Mark Donohoe. Welcome, Mark. How are you?
Good to be back again.
Mark, it's an area of intense medical research, and that is the human microbiota and the human microbiome. What we're going to talk about today, though, is the infant microbiota and how it develops from birth.
Isn't it fascinating? This is like what you must remember when I was a medical student, we were just coming out of those times where what we knew for certain in medicine was that breast milk was inadequate for the baby, and that cow's milk substitutes fixed those natural deficiencies that God or whoever she is put into breast milk. What we were also learning then was that the only way to have a good baby was aseptically with a caesarean, and there was the emergence of two parallel issues, both of which haunt us to this very day. Admittedly, the breast milk substitute is now much more a third world issue than it is a first world issue, but we still have the obstetricians cutting women open, believing that a sterile birth is a good birth and anything else is just messy, dirty, and not to be put up with by the profession.
This was the same era, though, where we had benzodiazepine receptors, and so people with anxiety or insomnia had a deficiency of benzodiazepines. Is that correct?
Well, I will tell you that I have to go right back in 1975, I remember distinctly in pharmacology that the bad old days of the terrible anxiety-relieving drugs, many, many different varieties, those days were gone. Pharmacology had developed the perfect drug, no addictive capacity, didn't disturb cerebral function, could be used at any dose, no overdosage effects, and it was now we can manipulate the minds and there are no downsides to this. And they were called benzodiazepines.
Whoops.
And you come forward a couple of decades and now the single greatest problem that we have is all the problems that we said would never occur. So you would imagine that we would one day lose the hubris, that every time we think we know better than nature, every time we think we have a good grasp of humans and biology, the only thing you can guarantee is fast forward 10 years and it will be looked on as the bad old days.
That's right. And so to microbiota, because we used to think, indeed, I argued vehemently with a colleague about this, that in utero, babies were sterile, and that if you had anything other than that, it was septicemia.
Yes.
Whoops, I was wrong.
Yes, but it was a good thought at the time. You know, the idea that microbes could actually leave the mum and migrate and turn up in places and start work even before birth was absolute heresy. And as you said, if doctors thought it had happened, we'd be putting every mother on antibiotics all the way through pregnancy to make sure none of those little buggers got across the placenta. So thank God we didn't know, or we would have tried to fix it. But it is fascinating that there's immune privilege to certain bugs that we grow up with, and they are allowed to travel through our system as if they were self. They are not considered foreign. They move to the places that start to colonise and start to modify the baby even before the baby leaves the womb. And that is just mind-blowing when you think of our prior ideas about the sterile womb.
I think we need to give a thought to just how many bacteria and even viruses and fungi there are inhabiting the human intestinal tract and indeed on us as well. Over 1,000 bacteria can inhabit the point of a pin.
All right. That's easy. I mean, that's easy for a bacterium. They're tiny little things, but just in sheer numbers, how they dominate the landscape of every human. We are big-celled creatures, and therefore we dominate in terms of volume and size. But when it comes to function, when it comes to every surface, we are outnumbered 10 to 1 on most surfaces, 5 to 1 on some, 100 to 1 on others. These microbes are doing a job on all of our exposed surfaces. Everywhere around the body, we have a layer of defenders that we never even realise. We think the skin's impermeable, the gut keeps the microbes out, we have mucus and mucin layers, and we have all of these mindsets. And then we find out that these are really creating environments for our fellow travellers, and rather than being terrified of them, the kind of Louis Pasteur came to this conclusion at his death, rather than being terrified of them, we have to understand that the 99.999% of these ones are not only just, they're not passive carriers, they are doing something for our health every step of the way. So we are symbionts. We do something for them, like go and find food and keep our skin clean and do these kind of things, and they look after the rest of us. And so we are their block of flats in a way. They look after us, they scrub us, they clean us. We think that we're having good thoughts, we're happy, we're sad. We never understand that the molecules that these little fellas produce get us to do activities. And we are their automatons. In one way, when you go for sugar, you may think I'm hypoglycemic, but in fact what's happening is the bugs that like sugar are saying, a bit of sugar would be nice at this point. And we relieve that itch, by making our little microbes happy without ever understanding that they're there.
So given that these bacteria inhabit every exposed surface, and indeed, some internal surfaces of the human body, let's concentrate on the gastrointestinal tract and how our infant microbiota develops. So one of the main ways of attachment is via certain sugars.
Yes.
Talk to me about what happens here.
Well, the sugars are found on the surface of all cells in the mucin layer. Pretty well all of the cells in the body have these sugars hanging off the side. We know the ABO system of the blood, your blood group A, B or O, but there is a tissue system which is the ABH system. And these are a kind of system of sugars that hang off, which allow certain microbes to attach or not attach. There's one variation of that ABH system, which we call Lewis non-secretors. It doesn't mean that our patients don't secrete mucus. It's that they lack certain sugars, certain glycans, that would normally protect against pathogen adhesion. And so the failure to produce the right sugars leaves us open to bugs that are pathogenic in nature, ones that our body normally doesn't recognise, bypassing that system which would normally hold our good microbes in place and protect us. So at every level across the body, these glycans and these sugar molecules are attracting, bringing along microbes. Those microbes are in fact consuming a lot of those sugars, and we provide the food for them, a little bit like parasitic organisms. They go and take those, live in us. We provide the sugars, the O glycans and the N glycans, and they go and live happily and protect us by metabolizing other sugars and providing us the energy.
So what about the mucin layer and the differences in the mucus layers throughout the intestinal tract? In the small intestine, you've got basically one layer. It's more liquidy, if you like, more fluid, and then you've got a rather thick mucus layer as well in the colon, which helps different inhabitants.
I think what I understand you're talking about is that there is, from the mouth all the way to the anus, there is a longitudinal huge variation in the environment we provide for the microbes, and so our mucus production and the mucin layer varies according, almost you could say we're coordinating the whole process, but the mucin in certain areas is highly supportive of a particular type of organism. We provide different environments all the way along the gastrointestinal tract so that the microbes that need to penetrate to get their particular sugars, their glycans from one area, are limited to that area. So in health, we have a mucin layer which is a very complex system of feeding microbes of a particular type at a particular place in the gut. There is the big transition, which we call the Ileocecal valve. It's probably more correctly the ceco-ileo valve. But there is an area where there is a massive change in the number of microbes from one area to the other. And right at the end of the terminal ileum, that's the very place that you have problems with B12 absorption. If you run into inflammatory problems with the failure for that ileocecal valve to work properly, you do get the B12 deficient and the nutrient deficient people. You also get a lot of gastrointestinal symptoms that come from there, and that could go all the way up to Crohn's disease in the extremes.
I haven't got any evidence for this, but I think it's very interesting that segmented filamentous bacteria, FSBs, which explode onto the scene in about week 3 from birth. And they're related to Clostridia, but they don't attack us like Clostridium difficile. There's sort of this, what do they call it, a pathobiont, they're termed. So not necessarily a pathogen.
Yes.
And they appear to prime the immune system as long as we've got copious probiotics to dampen an inflammatory response. and these inhabit the distal ileum. So it's kind of like, the appendix wasn't just for herbivores. It seems to be this area of intense immune activity.
It is true that in the New England Journal of Medicine at the turn of the century, surgeons regarded it as just one of those failures of God to keep the tonsils and to keep the appendix, and that was the God-given job of the surgeon to remove tonsils and appendices. And when the argument arose, it was, we surgeons are given God's job. His failures are our job to do, to tidy up humans. And that shows you a kind of level of endemic disrespect for what biology is capable of doing. The appendix in all animals that have appendices is a little sac. It's an insurance policy. When you get diarrhea and everything else is scoured from top to bottom and there's not much left in the bowel, you've got one little pocket that did not participate in that can re-establish the gut flora extraordinarily quickly. And so you do have a kind of perpetual sampling system that takes a microbial snapshot in real time, seals it off a little bit, takes another snapshot at other times, and the appendix has become a really, really interesting area of that, so that rather than it being residual and a kind of a nuisance and something
artifact
to get rid of, and good only for getting appendicitis, it seems like that is very much a stabiliser, so that once you get a microbiome underway, you keep a packet aside, like putting it in the safe. You just keep it off to one side, and when the need arises again, you draw back from that deposit box and say, okay, let's re-establish normality. And when we use broad-spectrum antibiotics, we're often dependent upon the appendix, so that when we change the microbial balance significantly, the fact that people get better, even after significant clostridial infections, is often a tribute to what we have retained, the kind of memory that we've retained of the original microbiome.
So talk to me about what happens with when you use certainly repeated courses of antibiotics in infants, because it's a huge issue of decimation, and it has been shown to affect the microbiota.
Yeah. I also have to tell a story that I saw a number of parents from Melbourne about a decade ago now. One of the hospitals there made it policy that a baby at birth, if they vary by more than 25% from the predicted weight, either too high or too low, should go on a 3-month course of antibiotics.
3 months?
3 months of a broad-spectrum antibiotic, because it would normalise the abnormal microbiome. Now, that kind of a percentage variation from predicted is never a good idea. The idea would be get the breastfeeding, get the vaginal birth, do the things that we know normalise that microenvironment. What happens at birth, what is critical to biodiversity of the gut for a baby, is that they get a gulp of the microbes as they exit the vagina. And that gulp of muck is what doctors were trying to stop happening. We believe this would get into the lungs. It was a terrible design flaw. The vagina was put way too close to the anus for any sane God to be able to stand up for that process. But that first gulp, that starter culture, is an amazingly important thing. What happens immediately afterwards is that the baby goes to the breast, and human breast milk is the most complex set of glycans of any mammal so far measured. What that means is we've got a higher variety of sugars, long-chain starches, all of these different types of glycans. Why is breast milk so diverse? Because it has a hugely diverse microbiome to start with, and for an omnivore, you need a whole range of different bugs. If you're a herbivore and a cow, you can get away with a very limited range of glycans in the baby's milk. And that's why children who are raised on cow's milk don't get the variety of glycans, they don't breed up the variety of organisms, and they tend to suffer the health problems of obesity and problems that are related to altered microbiome, even in the first 2 or 3 years of life.
So when you're talking about glycans, that includes these monosaccharides like fucose, mannose, uramic acid, things like that, but other food components as well.
It does. It is basically all of the starches, all of the carbohydrates, all of the polysugars. So in nature, cellulose is a glycan. It's an indigestible glycan for most of us, and yet recently, what does the Microbiome Project turn up? It turns up that some humans have some bacteria that break down cellulose, that those people can actually utilise cellulose as an energy source, whereas most of us package it up and that's what makes our poop. So the glycans are the broadest group. If you think of the fats, carbohydrates and the proteins, the broad sense of carbohydrates, from the monosaccharides to the disaccharides, sucrose and lactose, all the way up to the starches and the polysaccharides, to the N-glycans and O-glycans, which are absolutely filling the mucin layer. They're all carbohydrates, CHOs. They are not proteins, there are no amines in there. They have the oxygen, so they're not the hydrocarbons of the fats. Yet the microbes down there take those glycans of all the different varieties and will produce, say, short-chain fatty acids, the butyrates and the short-chain fats, which are so essential to colonic health. So they take starches on the one hand, things that our body can do nothing with, and turn it into useful material for energy production for the host, for successful repair and management and motility of the gut. And those, while we think of them as sugars, they're not. What we as practitioners talk about as problem sugars is when you overload the simple sugars, when you overload the sucrose and the glucose and the fructose. When you put too much of the simple sugars in, you do get an overgrowth of the firmicutes, the ones that are the really good sugar metabolizers, and you can also get an overgrowth of the ones that we don't like, the fungi, the candida and the fungal elements, which when they overgrow can cause really serious problems with immunology. But that process of going from the gulp of muck to the breast milk breeding up a very, very broad culture of a microbiome happens in the first 2 weeks. And the failure to do that means that caesarean children, non-breastfed children, have a stunted microbiome that does not participate in that process you were referring to of let's organise the immune system, let's see what we can do to keep inflammation under control, keep a barrier for that not to get out of control. And you do see this in kids, not that they're dreadfully ill, but that they get recurrent infections, that they have growth problems. And the growth problem for many of those kids is that they're just fat. They're getting too big too quickly. And that's going to end up as obesity and type 2 diabetes later on in life.
I want to get on to a subject which is of great interest to me, and that's the FODMAP diet later. But firstly, I mentioned just before about the SFBs stimulating the immune system, and one of the biggest ways in which this is achieved is by the plasma cells making secretory IgA. Talk to me about the importance of secretory IgA and the mechanisms thereof.
Secretory IgA is a fun molecule. I mean, it's one of those ones that we do know is manipulated by things like meditation and lifestyle changes and happiness and smiling. Immunoglobulin A we take as the surrogate marker when we measure it in blood of something going wrong on the gastrointestinal tract or the respiratory tract. The mucous membranes, when something goes wrong, when an invading organism hits, you usually see the immunoglobulin A levels go up in the bloodstream after a while, but that's always a consequence of something going on with secretory IgA in the surfaces. And so the secretory IgA is, if you like, within that mucin layer, a first line of defense which just says, do not enter. If you fail with the secretory IgA and things are making it across that mucin layer, then depending on other areas of gut integrity, you start to get immunological interest from lymphocytes and the B lymphocytes, which are the antibody producers. And so if your IgA is doing its job, most of that never needs to happen. You keep the surface under control. I liken it to having police on there. It's not bad to have the IgA up if there's a problem to be solved. But what we do see is that the secretory IgA and the IgA staying high for long periods of time normally results eventually in chronic inflammation.
So this is an interesting way in which I view secretory IgA, is that it's a reaction, a normal reaction, but a reaction to the outside world, to potential invaders saying, listen, you're there, I know you're good for me at the moment, but just keep your distance. So it's kind of like fence palings on a fence, or the picket on a fence, correct?
All right. Well, I said a police force, but you're probably right. As long as that is not breached, the ups and downs of IgA are absolutely normal. There is competition with every single meal for the nutrients that go into the body. With every breastfeeding, there's a change in that microbiome that goes on for months while the whole process settles down. And the immune system's job, remembering 1 out of every 10 cells on the gastrointestinal tract is a suppressor lymphocyte. There are guards around there just looking at everything that's coming through, going, friend, foe or food, friend, foe or food, friend, foe or food. And it is a really difficult job to fix. And the Secretory Iga is, if you like, just that paling that says, generally, bad guys, stay on the other side of this fence, would you? And we will leave you alone.
But interestingly, and this is something I want people to be clear about it, and that, they might get into the idea that we're saying that Secretory Iga is therefore bad. No, it's not. No. It's a reaction. It's just a reaction to keep the fence. Yeah. And that is there for the good guys as well. For instance, Saccharomyces boulardii increases Secretory Iga. But that's not necessarily bad. That's just saying you're outside.
Yeah.
Don't come in. That's fine.
It's a recognition of a molecule.
It's a recognition, that's right.
And it is, maybe you'll change my metaphor now. The fence building is probably it, that if you've got a well-constructed fence and it keeps the dogs and cats out that would otherwise have squeezed through if the paling was too wide, then that's a very good thing. And having a good quality fence there is a useful thing. There is a point where things are getting through, where the Secretory Iga, it's like the fence has turned into barbed wire fencing with guards sitting on top shooting at everything that's going past. That's not a good point. And so you do see then immunoglobulin A levels rising to, let's say, 5, 6, 8 grams per litre.
That's blood. That's serum, right?
That's in the blood. Yeah. But that's saying there is an IgA production, which is an unusual enemy being confronted down there in the gut.
That's right.
And the Secretory Iga generally goes up with that. When Secretory Iga drops too low, we have a problem, and that is things start to move across that wall, and it's not that it's free access, it's just that what's the second line of defence is an inflammatory type of response. The IgA deals with it very, very easily, very, very straightforwardly. And what you don't want is for it to be too low, and you don't want it to run off the top of the scale either. And so we do read the IgA as, how's the fencing done? Is it reasonable? Are there any holes in it? Let's go and fix those holes. So as a practitioner, you can measure Secretory Iga if you get stool samples. You can measure IgA on the blood, in other words, the immunoglobulin A, and the combination of those two gives you a pretty good idea whether there has been a chronic infectious type of invasion. If there are bugs there that really are not meant to be there, and that's commonly parasites, blastocystis, fungal infections, if you're seeing those kind of things busting the fence down, then you go and have a look at that from another perspective. If you've got a fight with your immunology on the gut, that's never a good idea. An inflamed gut is not doing the job that it's meant to do, which is absorption, nutrition, and management of the microbiome. So
would you then combine that sort of Secretory Iga test with, say, a lactulose mannitol test?
Yeah, I'm using the lactulose mannitol test much more these days because my interest is what kind of porosity is there in the gut. If the gut wall integrity remains high, if the lactulose and mannitol are reasonably good low levels, then I have some confidence that the intercellular, the kind of binding between the cells and the tiny little, 5, 6, 8 angstrom holes that are normal there to get molecules through into the body, that they're reasonably intact. You can overread that test because that test is only useful for the small intestine, lactulose and mannitol. And we do know that there is gastric intestinal permeability, and there is colonic intestinal permeability. And probably we should move on to eventually having readily available tests, the sucralose test for the absorption test for the sucralose test, which is done for colonic permeability, and oddly, a sucrose test, which is done for gastric permeability. You give a dose of sugar, plain old sucrose, it should not get across, and if it does, it's getting across in the upper part of the gut.
Can I just interject there? You said sucralose test.
Yeah.
Now, we're talking about the artificial sweetener here, yes?
Yeah.
Because I've had concerns about this for quite some time, in that not only does it appear to affect the P glycoprotein pumps, but it's been shown to affect bacterial colonies in the gut.
Yeah. And the test is an artificial test, just as lactulose and mannitol is. You're putting a challenge on to find out if something that should not get across actually does get across. The sucralose is used because it normally gets through the small intestine without being altered. And then if it's degraded by any microbes down in the gut and you're getting the sucralose components coming through, that gives you a worry about just the permeability of the gut. What we're worried about is there's a lot of muck that's made when those organisms really get to work on the small intestinal contents when it goes into the colon, And that auto-intoxication concept, when you have rapid fermentation and rapid degradation of molecules, the colon is meant to be a secretory organ, not an absorptive organ. And so a lot of dirty work goes on there, and if you get too much of that kind of fecal microbial metabolic products, people feel very sick just from the colon. And, megacolons, chronic colitis, those type of things can be quite serious diseases. People can be extremely unwell, get chronic constipation or chronic diarrhea just from variations there.
So let's talk about constipation and diarrhea, and we'll take it back to the immune situation in young infants, for instance. What sort of therapies would you use?
For which, though? There are many variables here. For babies that are breastfed, that have had a normal birth, if there hasn't been use of antibiotics, if there hasn't been anything else, it is rare for there to be a real problem with the poo. Now, that's not always the truth, because mums do absorb antigens, and certain antigens do get across in the breast milk. And so it is possible that things that mum eats, even such things as garlic and high sulphur foods, many of the things mums eat, they know affects their baby's poop. And they know what gives diarrhea and what doesn't, and the mum pretty quickly works out, I'm not going to be eating broccoli this month, I'm not used to that. So babies depend a bit on mum for the antigens to get through there. There is a serious issue, and that is true food allergy, which is thankfully rare, but where you get an allergic response, the best known one is peanut, and the recent study that you will have heard is that the peanut, providing the peanut with the probiotics to a person, the probiotics do a lot of the work in modifying the immune response so that it does not overreact and does not cause anaphylaxis.
Now, 6 months ago, there was a trial showing that 1/200th of a peanut, in fact, no, it was longer than that. About a year ago, there was 1/200th of a peanat was used as immunotherapy. Mm-hmm. So I haven't looked at this.
Homeopathic peanut.
Homeopathic peanut. Don't start me. But has the results of that trial with using peanut only been compared with the peanut plus probiotic trial? And this was a Lactobacillus rhamnosus long code. I can't remember the code. I've never seen it before.
Yes.
And it's from China. It's C something da-da-da. Right. It's from the Chinese culture collection.
See, this is why I come. You fill me in with the information that I would never take the time to otherwise know. That obsessive compulsive mind of yours works well for me. The whole issue of peanut and anaphylaxis in food is an absolute mess in medicine. There are camps saying never a molecule, mums should never eat it, their grandmothers should never have eaten it, and there's others saying load up the peanuts. I think the sensible middle ground is probably going to become normalise the gut, provide enough of the antigen for it to become familiar to the gut immune cells and for the bugs in the gut to do whatever the magic is that they do to help present it. And if you want my bet, I think we're going to find that many of the gut bugs act as almost pre-phagocytes or pre-macrophages, that some of them take the molecules in, immunologically present it to the gut in a way that we would normally think of as a macrophage. Grab that, we know that's you, there's the little sign, that's the pathogen we'll go after. And I think if we can grab our real understanding of the gut, there are those kind of agents, many of them derived from the soil, that you will get in normal food that may do that job of presenting the antigen to make it safe and develop a good suppressor response to make sure that doesn't get out of hand. That's pure speculation, but it makes sense that when you give a probiotic and you give the antigen, you don't see the rate of reaction that if you don't give the if you don't give the probiotic.
So in this trial, they used the peanut immunotherapy plus Lactobacillus rhamnosus C whatever.
XXX.
C. Yeah. I think it was a 4 or 5 numeral code. That's from China. If you can't get that, and I haven't seen it in Australia yet, what would you use? Would you use just Lactobacillus rhamnosus? Or would you use multi-species probiotics?
Oh boy, you're opening up a wound here because...
Against any evidence?
Yeah, no, it's not.
Or
just
not against
that.
any evidence, without evidence.
When research is done, how much can you extrapolate from the strain used to normal life, and how much can you extrapolate it to other products? In the world of drug therapy, there's a patent on everything, and something is used for a reason, and it's because somebody paid for that study. When you see results like this with a particular strain, the temptation is to think that proves everything that I said, which is if you just get the microbiome right, everything will be perfect. And that definitely is not true. But there are people who have never had antibiotics, who have peanut anaphylaxis and have been breastfed, everything has been right, and they go right through that same reaction. If you think of the probiotic as a drug-like protective effect, it is growing living organisms, but those organisms have a pharmacological action, and that pharmacological action is capable of suppressing what would otherwise be a rapidly escalating dose, then you have to at least start as close to that original organism as you can. And that comes right back to what we know of in complementary medicine, that part of the dampening of innovation is if you discover something like that, what's to stop someone stealing it and just walking off with it in another 5 minutes' time?
That's right.
Now, bugs are a particularly interesting one because just as we've got the genome that belongs to the world, and various countries argue that you can't patent it or you can patent it, the bugs that belong to the world do vary nation by nation. There is a whole ethnobugology of the gut that's yet to be done. The Microbiome project is the kind of tip of that iceberg starting to say, could this be true? But now we've got thousands of different cultures brought up on hundreds of different types of foods. And what we're worried about is first world kids who are the least vulnerable of the whole lot, and trying to apply it to third world kids to manage their guts. I have to give a small anecdote. I had children who were adopted by a family who came from Africa, and they came to me and said, we want these people's guts, we want it assessed, we want to know we've got healthy children. And they were. The healthy children, I analysed the gut, and I saw these microbes. There were monsters in the gut. And I said, we've got to get these kids on probiotics. Lactobacillus, bifidobacteria, all of these, the kids nearly died as a result of prebiotics and probiotics. And what I had missed at that time was I'm coming from an Australian doctor seeing normal guts for Australian children. These kids had grown up on a different diet with different bugs. The bugs that I saw as monsters were in fact very normal for them, and that's what the Microbiome project is uncovering. That variations of glycans in the diet, the high vegetable diets of many of the third world kids, breeds an entirely different microbiome, yet just as healthy as the Western microbiome, which we're largely breeding up on sugar these days.
So back to the probiotics, when do you find it useful, or when do you find it necessary to use single strain identified species of probiotics, or when do you find it useful to use a broad spectrum approach?
There are guidelines for this. There are some times that we don't want lactic acid production, that we don't want to acidify the bowel, and so the lactic acid bacteria are ones that you would tend to avoid at that time. I think that there's a confusion right at the moment. We have the problem that many of the firmicutes, many of the sugar metabolizers, we're not sure about what part they play in obesity yet, that they may be very normal for babies, and that's where we've got a lot of data and protection. But when you go and just translate that to adults, we've got a problem with obesity. We may be, in fact, giving some strains of probiotics which are obesogenic. These are things that have yet to be worked out. I don't go so much for individual strains, but where there is clear evidence, clear clinical evidence that there is something, so for example, the peanut and that strain of the Lactobacillus rhamnosus, I would be as close to that strain as possible before I had any confidence that something as important as anaphylaxis was likely to be able to be managed. When it comes to restoring gut flora, you got to remember the baby originally got a mouthful of poop and then a bunch of breast milk, and they did the rest of the job themselves. So I think what's important is keeping biodiversity in the gut open. That has a kind of a thought associated, and that is if you're going to use probiotics, use variety, use width, rather than try and be too specific. When we know in the future what bug we want to do a particular job in a particular illness, I'll be all for that. But right at the moment, what we can do is we could go broad and say, there are living foods. You could get kefir and you'd have unknown strains in that. But what we can guarantee is that the strains that are in the probiotics, say for at the end of pregnancy, that last 3 months of pregnancy where it's important to have that variety, we want the first mouthful to be the best mouthful that the baby can get. And if you can emulate the strains that are normally there in healthy babies, and you can get the mum to be secreting those, that's the best we can do.
And I want to add a caveat in there. for the listeners, and that is at no point should you be treating peanut allergy with probiotics alone and not under medical care,
Yes.
close medical supervision.
Yes, I think anaphylaxis management is probably a very specialised area.
Yeah.
I would not even do that in my
So
own
let's-
doctor's surgery.
Let's just put that in the area of interest.
Yes.
But back to breast milk, there was some very interesting research. Indeed, it was a group of researchers that secondarily described the enteromammary pathway and the use of certain probiotics. They used certain strains to manage conditions, for instance, like mastitis. Indeed,
Yeah.
better than antibiotics. But what I noted in the papers that they wrote was that at no point did they use the gold standard antibiotic that's used in Australia for mastitis, and that's flucloxacillin. They used cloxacillin, which is different, and also the amoxicillin and clavulanic acid, blah, blah, blah.
Right.
So can you comment on the practical implications of using probiotics for management of mastitis and indeed pregnancy, healthy pregnancy?
Well, not just management of established mastitis, but getting a probiotic approach to minimising the risk of mastitis after the birth of the baby. I mean, this happens very regularly. I think it is worsened by our medical tendency to give the mum a shot of antibiotics if they show any sign of group E streptococcus on the vaginal swab. That trying to be helpful is in fact not being all that very helpful. It was a real surprise, wasn't it, to find out that bugs inside the colon can make their way around the body and can modify disease risk in other areas. So my approach has honestly been keep the mum's diet with high variety in the last 3 months of pregnancy, introduce the probiotics, try and keep the microbial flora up to scratch with all the stresses and everything at the end of pregnancy. That is a vital time for mum and for the first month or two after the baby's born to minimise the risk of mastitis. And if you could stop us using antibiotics by just giving us the strains that worked, that would be magic. At the moment, we still lack the ability to be able to fully prevent mastitis.
I think it was really interesting research. Indeed, I think it was the second trial they did where they used Lactobacillus salivarius, Keck strain, 5713 or whatever, and they got better results than the antibiotic.
Yeah.
I thought that was rather encouraging.
It is. It is. And it may give doctors a bit of a better way of starting to think about this. What's happening with the Microbiome project is it's on doctors' minds. Probiotics are now a hot topic. All the research that's being done links back over a whole 100 years, probably forever actually, going back 5,000 years. But once you get a lot of money behind it and a lot of technical papers, doctors become confident. And what we do know about doctors in general practice is when they're confident in the treatment and the patient is confident in the doctor, diseases like mastitis, diseases like chronic diarrhea go away. And that's an important movement. Giving antibiotics was always a bad idea. Giving probiotics is always a good idea. The aim is to do no harm and to get that benefit.
Dr. Mark Donohoe, thank you once again for taking us through some practical implications of the use of probiotics in pregnancy and early infancy. I thank you so much, and I'd love to continue this on a second podcast on this topic.
It will be my pleasure.
This is FX Radio. I'm Andrew Whitfield-Cook.